We start our class, so no more swearing. And here we are, all right? So, all right, so first of all, I have your first exam, and I'll hand it back into the class today. And so that'll be a focus this week. And so we're going to jump right into chapter 17 now. It's cell cycle, and we've been foreshadowing it really the whole semester. So this is going to be a big, important chapter for us. And this is a chapter where you'll know you have some background from intro bio and maybe some other classes about the cell cycle. So it's going to be about the eukaryotic mitotic cell cycle. And that fits right into this because we're in this section, the fourth section in your textbook. And it's about cancer biology. And cancer is typically thought of as an out-of-control cell cycle, right? Like the woman with skin cancer, we learned about her. So we're going to learn more about, well, what is being stimulated by MYC. Right? And how does MYC drive the cell through the cell cycle? So chapter 17, read that. And then you need to start dealing with your term paper, so we'll talk about that in a minute, just to talk about what is that about. And then go over exam 1 answer key videos. So I made an answer key video, and I'll post it on Brightspace. And it's a video where I take the exam and I talk through it, because I take it if you haven't had a class with me, this is how I grade exams. When I grade exams, I don't hardly ever put any comments on them, either positive or negative. I just mark points. And then at the end of your exam, there's a score. There'll be a score, a circled score, and that's how many points you got out of 100. That's your score. Okay? So after I grade the exams, I take the exam, and I'm just talking through the exam as I'm answering the questions, and that allows me to provide comments for everybody. Lots of times, students will get all the points on a question, but yet their answer wasn't really that great. And it could be better. And so there's lots of learning for everyone, even if you got lots of questions right, because a lot of times they could be better. And we really want to learn this stuff because the final exam covers the whole course. And also, the second exam, this material builds on the first exam. So you'll see, we're going to go talk about a couple more receptor tyrosine kinases for the second exam. And so we really want to be solid on that material as we go forward. And so in the, and I'll talk a little bit more about that. But then we have an assignment. And the assignment is to go through the answer key video, go through your exam, identify 10 points that you either got wrong or that could have been better answers. So, just how could you improve your answers? And just write better answers for it. That's not going to change your exam score or your exam grade. It's a new assignment. It's just a different assignment. And the purpose of that is to get everybody to look at the answer key video. The answer key video is not perfect. There'll be questions you have afterwards where you're like, I still don't understand why I lost a point, and just follow up with me. You're not grubbing for grade or anything like that. You're just trying to learn, right? The whole point of the exam is to help you learn, right? Pause, see how you're doing, like that. Okay? So you want to do that too, okay? So that's where we're at on that. And so let me talk about this, and this is, you can't read it, but this is just everybody's name, and a while ago I shared 2 Google Docs with you, and that's where you need to do your work for your term paper. And there's a video I posted on Brightspace of how to do the term paper that should answer just about all your questions, and then our syllabus describes what the term paper is about. And it is, you pick a receptor, and it can be any receptor, and you write a term paper about it. And there's guidance in the syllabus about what the topics need to be that you cover, and you just go after it. And then the video explains, like, well, how do you write it, what's the format, and what's the sources like, what are the sources like. The reason I make these documents is because it's a way to reduce the likelihood that you would use AI to write your paper. You're welcome to use AI as a source. That's what Google does all the time now, right? I put up here, it's Google something and it'll give you an AI summary. Yeah, you're welcome to use that. You have to refer to that as a source in your bibliography. You can use any sources. You could use this course, some other course, our textbook, some other textbook, anything on the web, and you just have to list it. And I described that in a video. But what I don't want you to do is just have AI write the paper. So if you paste chunks of writing into these documents, I'll just assume that's probably just AI you had write. and you shouldn't do that. And I won't challenge you and say that's cheating. I'll just like be not going to give you a great grade and be impressed with, you know, if you paste in a bunch of stuff. So do the work in the documents. The documents are keeping a track of the editing record, right? So I can just glance at that and see, I see, you know, you really worked on that paper. So again, that's that. Now, if you say, oh, I can only write it out, well, I say in the video, take pictures of that, right? And just insert those into the documents, right? If you really want to physically, or map it out, right? Maybe you have a way you write papers where you make a big, you know, poster and get it all laid out, like a concept map. Go for it. But you can insert that in to show your work. Now, it's not a huge, stressful thing, but also it's not really likely that you're going to want to do all this the night before the paper is done. Some people can do that and be writing in it, and I can see, wow, you know, all the editing took place in the last 24 hours, and there was just continuous editing for 24 hours. You know, some people do that, and I'd be like, that's cool. You know, and I'd be like, wow, right? It's kind of like me peeking into your life, and I'm like, oh, wow. You know, you can do that, but it's better to get started. And some students have gotten started, you know, pick a receptor, blah, blah, blah. If you pick a receptor we already know some about, I'll know that, you know, and you just have to write about it and build up from there, you know. Or if you pick a receptor that I know we didn't learn about, then I'll know you started from scratch, right? So don't worry about that either. You know, pick whatever you want. I'm not worried about it. And think of it as another way to get points for this course, because some of you will find today, oh, you didn't do as well as you wanted on your exam. So I think that might be the next thing. So see, this is a score distribution. It's not from our course. It's from an intro-bio course, but it shows you what I do. So what I do for grading is that I just take that circled score you have and I make an anonymous distribution, and I just graded the exams and I see the distribution and I just look at it, right? So this was an intro-bio course that had 80 questions. The highest score was a 74, and I just say, well, those people got A's, A minus, B plus, like that. It's not based on a 90, 80, 70, 60 cutoff. I never do anything like curving it. I remember really well when I went to college and how much time my friends and I spent talking about how the different professors graded. And this is the best way that I can give you the best grade I can give you. And that is, this is anonymous, right? So there's a student that got a 63. That was their circled score. They got a B. So you'll see when you get your paper that you have a score. And then at the start of the answer key video, there's a distribution for our course. And you'll be able to see, and you can see why this person got a B, right? You know, they're clustered with other students that I thought also got B work and less than a B plus and more than a B minus, like that, right? So that's how I assign grades, like that. Again, it's not a curve or anything like that, or a 90, 80, 70, 60. I'll just say, I just think those are stupid. And you know, I could talk for days about how stupid I think a lot of grading systems are. You know, and so this is the fairest way I can grade you. Also, you know, I've taught this course a bunch of times. I've taught other courses, you know, and I see this work, and I'm like, that's A work, that's B work, that's C work. And it's all anonymous. You know, I'm not looking at you and thinking about you. I'm just looking at how the scores work. At the end of the course, I will have, and it's described in the syllabus, I just have the first round is just a calculation of what your final grade is. But then I start looking at you. Okay, now here's, you know, math. And I'm like, okay, and I start looking at it, and I'm thinking, and I look at it. One thing I always look at is if students can improve their scores during the semester. So if you didn't do that well on the first exam, don't worry about it too much for a couple of reasons. One, if you look at the syllabus, I think this exam is worth like 20%. The next exam is like 30%. The final is like 35%. So the points go up. And also, I super respect students that can increase their scores during the semester, because I think that's the whole point of college, you know. Here's another instructor with another topic. See if you can learn how to learn in that class, right? See if you can learn how to be a flexible learner. So that's what this is all about. So if you didn't get as good a score as you wanted, start working on your term paper. And then here's the other thing, but I've talked about this all along. The big focus is on what happens here in class, right? Learn that first. Our book is overwhelming. It would be a big mistake to just read your book one page at a time and think that you're really tackling this course, right? If you didn't have the text, textbook, you'd still do pretty well. Now, to understand what happens in the class, you really need a textbook to help strengthen what we just talked about. But because of that situation and our limited time, there are lots of things on the exam that just came up one time in our class. That's a big reason why I record it, is you just have to be getting notes on all of this stuff, right? And if you didn't get everything covered in the class meeting and you were here, just go home and watch it again like that. And organize it, try to pull it together. This is the format for the questions. There's a range of difficulty in the questions. You know, I talked a little bit about that on the video, because there has to be a range of difficulty in the questions, but none of them are trick questions. And I do some coaching in the video too. So not just answering the question, but I say, look, this is how you break this down. This is how you kind of, like a lot of times you have a kind of writer's block. You're kind of, oh, there's a question, I don't know that, I don't know that, I don't know that. And I took exams, you know, I don't know that, I'm stuck, and I just move on. Well, if you start doodling, a lot of times that can help break up your things or bring you back to, oh, it's about you know, a cell receptor, you know, yeah, we learned that pathway, and you can start sketching it out. So that's that, you know, we want to follow up on that and move forward. Okay, so here we go with cell cycle stuff. Oh, let me just pause and check in to see if anybody has any questions at all. Somebody might have this question. Oh, Sam, when is your next presentation? The third talk you're going to give about CAR T-cell therapy.
Thursday, Dave.
This Thursday, this week.
This week?
Yep. So here, let's just introduce that. So Sam's going to start our class and talk about that CAR T-cell therapy. And remember what he came to talk about so far? How to build a CAR, a chimeric antigen receptor. It's a protein, but you wouldn't study or work on the protein. Well, you wouldn't build it, the protein. You'd make a DNA gene. And so there's a gene. So we learned that. And then the second visit was, well, it's a receptor, and it activates several signal transduction pathways. So we looked about that. And the next visit is what does the T cell do when that CAR receptor is activated? And so that's what we're doing here in chapter 17. The cell cycle is a cellular response. In response to PDGF, the skin cells go through the cell cycle. So there are lots of different things cells can do to respond to a signal. The CAR T cells, we'll learn on Thursday, they go through the cell cycle, right? If they get activated, if they bind to the cancer cell, they're like, I'm doing the right thing. There should be more of me. I should divide and increase that population. There are other kinds of responses. We've already learned this concept with regard to cellular response, that cellular responses, like the cell cycle, those can include pathways that lead to changes in gene expression, and those are called genomic responses, and or changes that don't require gene expression changes, and those are called non-genomic responses. So here we have this, when the cell cycle is stimulated under a scab, it's a genomic response, because we have to have new gene expression, and we know that already. MYC gets activated, and MYC is a transcription factor, and so we'll learn about that. Sometimes it's new gene expression. There are ways to stimulate the cell cycle without having new gene expression, non-genomic responses like that. And so that's what we're talking about. And so here's just a quick look from the book at the cell cycle. And you'll see that the book, once again, has a lot more stuff than we're going to learn, and in a different order than we're going to learn it. So we're going to emphasize some of the stuff from the textbook much more strongly. So here it just shows this. Here's the famous cell cycle that has these cell cycle phases, so G1S, G2N. And this is the eukaryotic cell cycle, so it's not about bacteria, it's not about prokaryotes, but it's about a lot of cells. All plant cells, all animal cells, and all single-celled eukaryotes have this same cell cycle. It's called the mitotic cell cycle to distinguish it from the meiotic cell cycle. And we'll talk a little bit about the meiotic cell cycle. That's the cell cycle that's used to produce sperm and egg. in our bodies and all other animals and plants. And so this is going to be regulated. And a big part of what we'll do is we'll drop down to the molecular level and look at the molecular mechanisms of regulation. And there are some famous points in the cell cycle. And by far the most famous one is here. And we'll see the name for it. So there's a G1 into S, and there's going to be a cell cycle point there that we talk a lot about. And that's the one that MIC regulates, and that's really by far the most regulated one. So we'll name it, and we'll talk about it. Then, during S phase, when the DNA has been synthesized, if there's some errors, the cell cycle can stop. We won't talk at all about that. Then, at the G2 to M, so this is at what people would say toward the end of the cell cycle, where the cell is getting ready to think about dividing. And before it divides in M phase or natural mitosis, it can stop here due to DNA damage. And we'll learn about this. We'll learn about this one first because it's a pretty simple story. It's a nice package that you can be like, okay, I got that. Pretty straightforward. Over here, this is the place we really want to study because this is the most regulated place by far. But things are complicated over here. So our first stab at this will be here, and we'll really set the stage for moving over here and adding the complexity. Then, just one time, right now, we'll mention this point, and it's called the spindle assembly checkpoint. And it happens in M phase, and it's when the chromosomes are trying to line up, so you can see them under a microscope, and they're lining up, and there's some problems with them getting lined up. And the only reason we're going to mention it in our course is the spindle assembly checkpoint is often defective in cancer. So when a person looks at cancerous cells, they very often have the wrong number of chromosomes. And that just makes them more messed up. And that's it. So when you look at this, there are really only 2 points in the cell cycle that we're going to talk about in this chapter. First, that one, the G2 to M transition, and second, our favorite one, the G1 to S transition. Okay? So that's what we're looking at, and we got to drop down to the molecular level to talk about the little like molecular switches that regulate those transitions. Here's what I show in introviolet, and to start out with, there are some people who are really into the cell cycle that refer to the G1 transition, that will be our favorite. They call it a control point, and a control point is considered where the cell looks outside of itself to say, should I go forward or not? So that's where PDGF would come into play. The cell also looks internal to say, am I ready to go forward? The analogy for this G1 checkpoint, that control point rather, that we'll use is it's just like a traffic light. And if it's red, you just have to wait. And when it turns green, you can go. And we'll just talk about that analogy a bunch. MIC is going to turn the light green. Okay? So that's a checkpoint, and it has names. The most famous name it's called start. And that's because in animals, it's almost always the place where the cells stop and wait for a green light. And when they have the green light, they go through the whole cell cycle and wait again. So start is a great name for that. It's also called the G1 to S transition, because really, when the light turns green, you're going into S phase. You can kind of see where I use Photoshop to move that little red barrier from here to there. And our book shows this, and it's very common that books show that the G1 to S transition is a little bit before you get to S. It's really not. We'll see that when you activate and turn the light green, the cell starts to try to make DNA and copy DNA right away. It's all about that. The light can be red for a long time, and we'll talk about that. But when it turns green, it's like I'm heading to the cell cycle. It's just too much for people to believe that when the light turns green, you're just rolling into S phase, but you really are. So a nice name for it is G1 to S transition, because when the light turns green, you're heading to S phase. Because of that, red light there, cells spend a lot of time in G1. That's really where the cell does its living in our bodies. Almost all our cells are in G1. So that's that. Over here, what I like to say is you can think of the cell cycle as like doing laundry in a washing machine. And you know, it's kind of famous that if you don't close the lid on a washing machine, it will go through the wash cycles, but when it's going to spin, it waits. And it's waiting because the lid is up. And it would wait forever. And if you lower the lid, it moves forward. And that's the G2 to M checkpoint, and it's a DNA damage checkpoint. And we'll learn about that. And so when the lid is up, it just means there's DNA damage, and we're going to wait to fix it, because it would be foolish to go into M phase and separate into two cells when there's some DNA damage. That would be a dumb mistake, so let's just wait. It's a nice analogy because in the case of G2M, it will just sit there like that. And whatever, who knows why the lid up is a reason for the washing machine to stop. That's right, babies fall into it, cats get in there and you know, they're like, get in there, right? And just take a nap, right? So let's just wait, yeah? Okay? So that's the G2M checkpoint, and just like that. And it's called a checkpoint because people refer to checkpoints as when the cell's looking internally, right? The washer machine is looking internally. And then this, again, this spindle assembly checkpoint is that really odd situation where if the load is unbalanced, it will shake the washing machine, but if it's really unbalanced, the washing machine can just stop. And it won't go until you fix things up. So that's that M spindle ascending checkpoint, like that. So nice analogy because in that analogy, PDGF is your hand. So the cell cycle is a washing machine, and it is a control point is when it's waiting for an external force, like PDGF, and that PDGF pathway to be the finger that pushes the button. So you can also think of start as like the button that starts the process. And if everything works with the washing machine internally, that it passes through all the checkpoints, it'll go all the way down through, and then it sits there because your clothes are done. So it's a really nice analogy to think about. And so again, control point is an external force that's going to activate it. We'll see that when that G1 to S is a little bit complicated because in addition to that external control point, there's actually a DNA damage checkpoint there too. So again, it gets a little bit complicated when we get over to that. So we'll untangle that as we go, okay? So this woman who had cancer, it's all about her cancer cells are not waiting. They're going through they're going through the G1 to S transition, and it's because there's some rate limiting step that she is messing up. And we already learned from our first exam, the mess up is she's making PDGF in her skin cells. And her skin cells release PDGF, and then they just turn around and bind to the PDGF receptor, and they stimulate the cell cycle. So her skin cells are not waiting at the G1 to S transition. It's like the washing machine just keeps doing the same load of laundry over and over again because it's poking its start button over and over again itself. And it's just out of control. And you're like, oh my God, look at my electrical bill. I don't know what caused it. And you're like, wow, take a look at your washing machine because it's been working constantly. That's just what's happening to her in her body, okay? All right, so here's another way to envision what we're talking about. So here's our cell cycle chapter 17, and our big focus is that G1 to S transition start, control point, and here's chapter 16, and it's the signal transduction pathway that's leading to that. And we've already talked a little bit about the connection, about what MIC activates that's going to regulate the cell cycle. But we're literally joining those together so that we could start with PDGF and have the cells be stimulated. This is typically a really complicated pathway because there's a lot of checks and balances, there's a lot of integration happening, because the body is trying to be really careful not to mess up. And so we do have this one example that we can talk all the way through now, from PDGF to we'll be able to talk all the way through stimulating the cell cycle and driving it through S, making the green light go, like that. And that's true, but it's almost it's not an oversimplification. It's just that most often it's complicated, more complicated than that. That's fine. It's a framework that you can go on and add knowledge to. Okay, so that's where we're at on that. Here's just a figure from chapter 17, and it shows this. Growth factors like PDGF lead through the RAS-RAF-MEK pathway to get activation of ELK, and then ELK is going to cause activation of gene. In our case, we talked about MYC, and then MYC is going to stimulate the synthesis of these cyclin proteins, and these cyclin proteins are the rate-limiting step, in this case, under our skin cells, to turn the light green. So we increase that rate limiting step, and it's just we need more cyclins. If we make more cyclins, we'll go. In the case of our book does this foreshadowing too. It's foreshadowing some proteins that we'll talk about in chapter 19. And so it's just mentioning some other proteins. So it can activate MYC under the skin, but these growth factors can activate the cell cycle in a variety of different cell types. And they don't always use MYC. They could use some other transcription factor in some other settings. So again, it gets complicated, but we'll see that we'll add those proteins just a little bit, because the transcription factors in those cases are also famous oncogenes, like MYC. Yep. So again, we're just looking at this a little bit to set the stage of what we want to study, because we've already leaned into it, and then we're going to go over to the other side of the cell cycle and talk about the G to the M transition. All right? So let's just look at the cell cycle a little bit to add a couple of terms to it. So here's a couple of terms. So up until, before 1980, scientists didn't understand the cell cycle, and all they could do is look at it under a microscope. And when you look at cells under a microscope, what you see are just two situations. Either the chromosomes are visible or they're not. They're visible because during mitosis, the chromosomes condense. And during the other times, they're decondensed, and the nucleus is there, but you can't see any chromosomes because they're spread out. So if you look here, we have two terms from the olden days, and it's still true today. There are really two phases of the historical cell cycle. One is called interphase, and it's made up of G1, S, and G2, and the other is called mitosis, or M phase. We already learned about the mitotic index. The mitotic index is measured looking under a microscope, and the mitotic index is a percentage. Count 100 cells of normal skin cells and determine the percentage that are in mitosis. You're just looking at 100 cells and seeing how many cells can I see the chromosome. And people have been able to do that for 150 years. So the mitotic index is an old measurement, and but it's still used. It's still used every time a person has a biopsy, a tissue sampling of a lump or a bump or a mole. The number one thing that doctors and the clinicians do is they measure the mitotic index, because the doctor is going to come back to you and say, I'm not worried about that. It's growing slowly. I think it's benign. They're talking about the mitotic index. Yep. So that's based on just looking under a microscope, okay, and seeing the chromosomes. And the other thing we want to mention here is this other term. And so over here in G1, I said that this is when cells spend most of their time. All our differentiated cells are in G1. Our muscles that have stopped dividing are in G1. All our cells in our brain that stop dividing and will never divide again, they'll be with you your whole life, but they're never going to divide again. They're all in G1. And because they're in G1 so long, scientists decided to give a special name, and they call it G0. And you can see what it's like is that the cell cycle is like a road map, and this G0 looks like a detour. Looks like the cells are actually going on another path. And that's fine, but in a course in cell and molecular biology, we have to say, yeah, we can use the term G0, that's fine. But you know what all your brain cells are doing right now? Is they're all parked at a red light, the same red light, waiting to go. And it's just hard for biologists to believe that's true, but yet it is. They're just stopped at the red light, waiting to go. And they're never going to go.
So differentiated, is that?
Yeah, differentiated cells that have stopped dividing forever, they're just waiting at the red light. Yeah? And that's important to appreciate because the red light, green light thing, it's just a biochemical thing. And the mechanism is just not there for the cells to advance.
What happens if you give them the mechanisms to advance? Do
They go forward, yeah.
Which is
Yeah. If to keep that answer simple like that, here's a simple answer to that. So again, sometimes we'll go over to the G to M transition to answer questions about start, because it's just a simpler story. But just listen to this. Anytime these cell cycles are regulated and activated, the cell just immediately goes into mitosis. So we can force the cell to go into mitosis at any moment, any time. We could make any differentiated cell just go into mitosis just by activating that. So it's all just about the biochemical regulation of that, red light, green light. Remember over here, we wouldn't really think of it as sort of a red light, green light, because we want to make it distinct. But it's like the lid is up, And if the DNA is damaged and the lid is up, if we just manipulate the biochemistry and pull the lid down, the cell will just roll through mitosis. And that's true over here, anywhere. If it's in the middle of S phase and we lower the lid, it just immediately goes into mitosis. And we'll see how that's possible. The proteins are in place for it just to roll forward. And so it's fine, and we want you learn the term G0, and everybody talks about it, but it's important to appreciate that the little car is not driving on a detour. It's sitting at a red light. And the people are like listening to music in the car and chatting, and they're just like, I can't believe the light has been red for 80 years, in this brain cell. And I'm just like, dang it, this is getting kind of tiring, right? Like that. Here's why we shouldn't talk about it as G0. Because when we are children, before we reach puberty, we have a bunch of cells that are in G0 waiting for estrogen or testosterone. And as soon as those show up, we roll through, the light turns green. So the light could be red for years in some cases, and it's totally normal. And then it just turns green, and the cell's like, nice, spring break, right? We get to go to Florida, and we're hanging out here all through our childhood, and now the light's green. So G0, and that's just the reality of it, and we don't want to criticize it because it's given a term because it's just remarkable. But the reality is the car is not taking a detour. It's always just waiting. It's just waiting, and the cell is doing all its living in G1, whether the car is really parked at the red light or another point along the pathway. So terms there, okay? So now a little bit more about when you can see the chromosomes, okay? And so that's famous, right? You can see the chromosomes. The chromosomes are condensed and visible during M phase. And we'll learn, when we learn the G2 to M transition, why they're visible. It's very simple. And it's true for every eukaryotic cell on our planet because these regulatory stories at G2 to M, they're the same. It's just a simple DNA damage checkpoint, and that's important, and it's been important since we were single-celled organisms. So you can see these chromosomes like that. They're only visible during M phase, right? Yes?
Can I ask you a question about satellite cells? So are they also sitting at the G1?
Yep, so satellite stem cells like we learned about in the muscles that are waiting to help if you need to get more muscles for any of a variety of reasons, they're just waiting at that. They're red. Everyone would say they're at G0. They may sit there for the rest of your life. If you're not injured, your muscles aren't growing, you're not working out, they can just sit there. They're fine. Yep, but people would say they're in G0. They're at that G1 to M transition. Yeah, most of their cells hang out there. Even our skin cells, as they're slowly growing, most of them are just sitting at the red light, waiting. the mitotic index is an indirect indicator of actually what's happening at the red light. Because when the light is red for a long time, there aren't many cells in the cell cycle. And those that are in the cell cycle would just be randomly scattered, like that. So let's just say you measured in normal skin cells, and we'll just make up a number. We'll say that 1.5% of the skin cells are in mitosis, in normal skin cells. So 1.5% are here in this phase, phase. There'd be some in S and G2, and that part is G1 also. Just scattered. This is just easy for us to count and see. Most of the cells would be at the start in G0, waiting at the red light. And so the mitotic index is just literally an indirect, easy way of measuring how many cells are waiting at the red light. Okay? Yep, okay? So next thing about that. Scientists can take a population of cells and they can do a method where the cells are swollen and then they're squished. They're broken open and the DNA gets spread out. And then they look at those blowing up cells under a microscope, and if the cell was in mitosis, they see a spread of those visible chromosomes. And so they look like that. And that's called a karyotype. And we've already used that term because that's how you can see, and have been able to see for years, those chromosomal translocations, like we saw in MYC and the antibody gene, in patients with Burkitt's lymphoma, or in the skin cancer patient with the skin collagen gene. Those genes were not understood until about 40 years ago, but 50, 60, 70 years ago, they could see those chromosomal translocations in cancer patients by doing a karyotype and saying, I see a problem with those two chromosomes. And that was diagnostic of the cancer type. So doctors and clinicians and scientists could talk about Burkitt's lymphoma for a really long time based on the visible chromosomal translocation, long before they knew anything about MYC or antibodies, like that. So that's when we see it. And here's just an interesting fact. Because of that, karyotypes are not visible, chromosomes are not visible during interphase. So karyotypes are only in cells that are proliferating. It would be logically impossible for you to do a karyotype of your brain cells, because none of them are in M phase. None of them have condensed chromosomes that are visible. So there's a limitation to karyotypes. That's just that fact. They have to be in the cell cycle, so they would have some cells in mitosis to have visible chromosomes to see a karyotype. So we learned about karyotypes because we learned about MYC and PDGF in those cancer patients, and this is just a cell cycle fact about those. Okay? All right. So here's another way that scientists look at the cell cycle, and it's an instrument that's called a flow cytometer, and a flow cytometer just causes the cells to flow past a little spectrophotometer electric eye, and so the instrument's called a flow cytometer. And they can be used for a bunch of different applications. But what they're doing here is they're just taking a population of cells and they're just passing each cell one at a time in front of the cell flow cytometer, and they've added a little dye, a little fluorescent dye that sticks to the DNA. And so the more DNA the cell has, the more the cell glows. And so here is the number of cells, and on the y-axis they're just counting cells, and then on this axis it's just really the amount of glow. And it's measuring the amount of DNA. And there's a little nomenclature here, there's 2N and 4N. That's the number of genomes. 2N cells are diploid, and 4N cells are tetraploid. And so if we just draw the classic cell cycle here, we have G1, S, G2, and M, and we'll learn about this in more detail, but G1 cells are diploid, one genome from mom, and one genome from dad. That's what we inherited, and almost all our cells are diploid because most cells are hanging out in G1 or G0. One genome from mom, one genome from dad. And then when they go through S phase, those get duplicated, so G2 cells are tetraploid. And so you see here, they're measuring the amount of DNA in each cell, and most of the cells are in G1. Some of the cells are in G2 and then going into M phase, right? And then there's some cells in S phase that are in between that. They're copying their DNA. So there's an instrument that can show us a little bit more of the detail in the cell cycle. And it's not a microscope, it's an instrument that is like a spectrophotometer. It's like a spectrophotometer that's going to be measuring the glow, and the instrument is just moving one cell through the spectrophotometer at a time. And flow cytometers can move 10,000 cells one at a time past electric eye, a spectrophotometer, a second. Like that, just thousands of them really fast, or a minute maybe, like that. They move a lot of cells really fast because that's used a lot. And we'll talk a little bit about flow cytometers and their use in cancer biology in certain types of cancer. But just another view, and our book shows that, and just to explain, what is our book showing? It's showing how the DNA content increases from diploid to tetraploid, and then the cells divide, it goes back to diploid, like that. So that's cell cycle look, like that. And then we can talk a little bit, again, because we foreshadowed this with the women with cancer, skin cancer, and measuring that mitotic index. So we learned about that, we asked questions about that on a quiz for that PDF video we did. And so let's just look at this to add to that knowledge. So here it is. It says mitotic, it's a research article from 15 years ago. Mitotic index is predictive for survival of dog cutaneous mast cell tumors. So they're just doing research and they're asking about this. So dog cancer, right? And it says here, so let's look at this data, right? It says the median survival time for dogs with a mitotic index less than 5% in those tumors was significantly longer, 70 months, than for those with a mitotic index less than 5%, 2 months. See that difference? So the doctor will, or the veterinarian will tell the owners of the dog, whoa, we took a little of the tissue out, did a biopsy, took a look at that cancer, and I'm worried because the cells are growing really fast. They will say, it's growing fast, I'm worried it might be malignant. And they're basing it, they could base a lot just on that for that type of cancer, right? Just really dramatic difference in survival rate. And this isn't talking about treatment or anything, it's just like that dog has got a really high mitotic index in that cancer, a specific type of cancer, and this is what we predict. And you hear that, for people, right? Where they're like, like that. Oh, my mom, for example, Lois, passed away several years ago from old age. But when she was old, the doctors found she had a kind of skin cancer. And they said to Lois, I'm not worried. We're not going to treat that cancer. That cancer's not going to kill you. Something else will kill you before that cancer, because it's growing slowly. So the mitotic index was low in Lois's skin cancer on her scalp, and they just gave her like some cream, like some lotion, just to rub on it to keep it from drying out. But they're like, we're not worried about the cancer. Really surprising. Like Lois was super confused. Lois is not a molecular biologist like her son. Like I would say to Lois, I'd say, I can help you with that, Lois, but first you have to tell me the 4 letters, right? She'd be like, because I asked her that question over and over again, and it's GACT, but Lois would just be like, is it N1? And I'd be like, no, like that. So, right? It gets, it's complicated. Here's the other thing. If you're going to college or spend any time in college, you will have relatives that turn to you for help with medical questions. So we're trying to set that up too, where you'll be able to not necessarily have the answer for Lois right away, but you'll be able to Google it and start sifting through it, right? Because medicine is really complicated, right? So, or for a dog, right? Tell somebody about their dog, okay? So let's take that a little bit further. So this is just some evidence, and this is what was published where they were using this to show the doctors in a hospital that make these judgments to explain it to your doctor that are called pathologists. And so this is just a chart for a pathologist to understand the likelihood of survival and the danger of a certain type of tumor. And we'll write the name out in a minute, gastrointestinal stromal tumor. So tumor in the intestine. And here they're just looking at two variables, the mitotic index and then the diameter of the tumor. You could think of these as being something like the polyps that doctors can find in a patient, and they have a lot more things they look at, and more things they look at, but they'll look at the mitotic index and just the diameter. So they're asking about this, the size, and then the mitotic index. And then they're just, saying, well, where was the position of this tumor in the digestive system? And then they're saying, what's the danger of it? Who should be followed clinically? And they're just using, in this case, the mitotic index. Is it high or is it low? And the size of the tumor. So that's humans, and with tumors like that. A certain type of tumor, again, not every tumor, but that type of tumor, they can get a lot of information from the mitotic index and just the size of the tumor when they removed it, okay? All right, so here's the word, gastrointestinal tumor, like that, all right? And now we say, nowadays, more and more, the doctors can add molecular features to this diagnosis and this prognosis. What is the deal with the cancer you have, and do we have something we can treat it? We learned that already with the breast cancer, right? That the first thing they do is not just a mitotic index and the size, but also, is it ER positive? Because if it's ER positive, we're going to give the patient tamoxifen tomorrow. If it's ER negative, we're not, okay? So here's an example like that, okay? And it says, these tumors are defined by an inclusion now of whether they have a mutation in another gene, gene for a protein that we're going to call KIT. And KIT is, and we'll have that next slide, it's another receptor tyrosine kinase, KIT. So most of them are by that. Look at that one. That's the PDF receptor. That can have mutations. So we didn't learn about that before. In skin cancer, that wasn't the story. The PGF was the story. But you could imagine, you could have a mutation in a receptor. PGF receptor could be a problem, like that. Yeah? Okay? What's going on there is the cells in our intestines, those are always growing slowly because those cells die and they get rubbed off just like our skin cells. They're exposed to kind of the, more or less the external environment, right? The food going through our intestines. And so those get rubbed off, worn out like that. So they're going through the cell cycle slowly. Most of the time they're sitting and G0, sitting at the red light, then it'll turn green, just like our skin cells. And what we could say is there must be, that KIT receptor must be involved in the normal stimulation of the cell cycle in there. So do we see KIT mutations in the skin cancers? I don't know. But if we don't, it's because KIT receptor isn't used in regulating normal skin growth. So what we're saying really here is it must be used significantly in our intestines. And the PGF receptor must be used there too, right? Like that. And you can imagine that because I'm sure you're aware of the idea that we can get ulcers, we can get wounds in our intestines that need to heal. And they could bleed some, right? And you would stimulate the platelets to stimulate clotting, and they would release PGF. So it's not surprising that there's a role for PDGF there, yeah, because there could be wounds that need to be healed, right? But looks like KIT plays an especially significant role. So here's just some more information about that from this Wikipedia page. The identification of a molecular basis for GIST led to the exclusion of many tumors that had been considered as GST previously. So that's a really important concept, is that doctors can use these molecular tests now to help categorize it. Do we have a good example of that? Yeah, breast cancer. The doctors can use molecular marker like the expression of the estrogen receptor to start categorizing breast cancers and saying, this is an ER positive breast cancer. We do have some drugs that can be used against it. So that happens more and more in cancer now. As you learn about, well, what is the molecular problem with this type of cancer, right? There's more and more likelihood that you'll be able to develop a drug against it, okay? So they started excluding, right? They were saying, well, these really aren't, we're not gonna call these other tumors of the intestine, gastrointestinal stromal tumors anymore, because they just don't have a KIT role. The KIT doesn't seem to be playing a role previously. And also the incorporation of much larger number of tumors that had been labeled as other types. So see, it really helps the doctor further categorize with better specificity what is the problem with this cancer, and what are we going to worry about, and what can we maybe use to treat it, okay? And so that could be the KIT receptor. And we won't learn too much about it, but it's a receptor tyrosine kinase. It has a growth factor that stimulates it, activates it, and it leads to the activation of signal transduction pathways like that, similar to the ones we've learned about. And here it's showing. So again, we're not too worried about the growth factor. There's KIT, it's a receptor tyrosine kinase, and look what it activates. It activates PI3 kinase and AKT, and it makes a cellular response. Then it also activates RAS, RAF-PEC, or HELP. So KIT.
What is the SOS?
Huh?
The SOS?
SOS, I don't know.
It's kind of fun.
Yeah. See, there's a sarcomology domain. So it's that intervening protein, and those change in different cell types. But it's the classic RAS, RAF-MEC, or HELP pathway to stimulate proliferation. So one receptor stimulates two things, and this is our first look at this, because this is foreshadowing our next chapter. Just about any time a cell is stimulated to proliferate, through one way or another, it's also going to be told to either kill itself or not. And so apoptosis is the focus of chapter 18. And apoptosis is a kind of cell suicide, literally. The cell just is fine, but it's just like, I'm not doing the right thing, and I'm going to destroy myself. Yep. And so in this case, what the growth factor is stimulating the cell, saying, go ahead, go through the cell cycle, and don't die. In some cases, growth factors or other pathways stimulate both. Because any time a cell gets stimulated to go through the cell cycle, it should worry if it's doing the right thing. It should worry. It should be like, oop, 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 oop. I don't know if I want to. Because what if it's out of control? So that's a big part. And so we'll see that in chapter 18, right? And KIT is a good example of that, So we go to different places in our body for these examples, right? Skin, intestine, right? Move to some different places to learn some different concepts. They're generally true. When a cell goes through the cell cycle, it's worrying if it's doing the right thing, and it might increase the likelihood of going through apoptosis, cell death. That's chapter 18. So chapter 16, the receptors and signal transduction. Then 17 and 18, these are cellular responses. Stimulating the cell cycle is a cellular response. Regulating this apoptotic pathway is a cellular response. In general, cancer are cells that are growing out of control and failing to die, failing to go through a self-suicide type thing, where they're just like, I'm not doing the right thing, and I'm just gonna wipe myself out. Yep. And so that's a nice example too, right? We put as many things as we can into each of these examples. So KIT. KIT is cool. KIT is a receptor tyrosine kinase. So we add it to our list, right? For our first exam, we had those that we learned about from chapter 16, and there are 58 total receptor tyrosine kinases, and we learned about PGF receptor, we learned about the insulin receptor, because that activates FOXO, and here we just learned a little bit about KIT. And these diagnostic tests really help categorize cancers. You have a problem with your KIT receptor, and so we're going to say you have a gastrointestinal stromal tumor, and we have some differences in your prognosis. You know, do we need to treat this? Are we worried about it? And it gets added to the other data. It gets added to the mitotic index and the size of the tumor, and then what is the molecular lesion, they'll call it that, mutations that cause that. So we just put it in there. And we'll have one more that we'll add in chapter 19. When we get to 19, that's our last chapter in the book, and that's more about cancer biology generally, where the authors try to pull it all together, okay? So that's that. Let us pause here and do a little concept chat action. And if you haven't talked to somebody, you know, there are plenty of students you haven't talked to in this class yet. You can go say hi to them and say, what's the deal? Are you hoping to graduate? Something like that. No. What are you doing for spring break? Spring break is coming.
Your book? Isn't that brand new?
Yeah, yeah.
Chromosomal translocation.
Yeah, just get ready for a chromosomal translocation, yeah. Just glue this onto another one.
So I'm Ian, nice to meet you.
All right. Yeah. So, yeah. So in some cases, we have an uneven number of students, but yeah, and then when we do, you have to have 3, yep. All right, so 3 things that we talked about that you thought were important today.
So that it's really not that far in G. It's actually like right before. So it's in C, but it's like right before the X. So he just Photoshop, I love that. And then
so stem cells, there's like a separate factor out of G1 that's a gene.
I'm so confused on if differentiated cells can technically undergo a cell cycle, why do we even need satellite cells? That was bothering my head for a while.
Yeah, beats me.
Because like maybe it's something we can do it out loud, like our bodies just don't want to do it, but it's just seems like such a waste. Like why give up the ability to go to my brain? Like why? Not how, why. He doesn't like my why questions. You don't like my why questions.
Oh, I like all questions. That's false. I disagree.
I love the SOS.
It really is.
Yeah, it's like kind of, but just so that actually I was like, oh, and then we have a new friend, Kit, and he is another type of kinase, right?
Where was it?
No, Kit is, yes, another type of kinase. I know that I've been tested for it with celiac disease.
You have to
pretty
run
common.
your dad is copying itself.
Like, to have an issue with it. So my brain was so far gone, I need to rewatch this, but you make me sad when you talk about dog cancer.
Oh, well, yeah, and that's...
They just removed a cancer not from Maisie, from my childhood dog.
Sure, yeah.
With the bucket margins and everything, so.
Right? Yeah.
But he's 14 years old.
Yeah, yep.
I've had him since I was in junior high.
Yeah.
How's my goal?
So we'll just keep rolling on this and for another minute, and then we'll talk and check in. So let us check with Rustam. Rustam, what's one thing your group talked about?
We talked about the 2 variables of this being size and mitotic index.
Yep. So that's a starting point always, right? The doctors have been able to work with that for 100 years because they see the mitotic index under a microscope and they see the size of the tumor, right? So that's always done. And then we add molecular details to it, like that. That's great. Oh, and what's another thing your group talked about?
Flow cytometry.
Yep, so a flow cytometer is an instrument, and you've all used spectrophotometers, so you can think it's a specialized spectrophotometer. And what it does is it just controls flow. And you have to have cells that are in liquid. So if it's tissue, you would actually have to use some enzymes to break the tissue up into individual cells, and then you put the liquid into the flow cytometer, and it just starts pulling the cells through a tube, and the tube gets narrower and narrower. So that by the time it gets to the spectrophotometer, the tube is so narrow that only one cell goes past the spectrophotometer at a time, and it's just reading the absorbance. And we added a dye that binds to the DNA and glows, and the more glow, the more DNA. And so it just is reading for each cell the relative amount of DNA. Yes, Justin?
I'm assuming that dye is really kind of dangerous to work with if it binds to DNA.
Yeah, that's a good question. And the answer is the dyes that have been used were dangerous because they could cause mutations. But now, organic chemists like all of you have developed other dyes, organic chemical molecules that are not dangerous, even though they still stick to the DNA. Yep. And also, I should say that the cells that go into a flow cytometer are not going. They're not going to be used again. They're going into the trash, like that. Just a side note, there are flow cytometers that are called cell sorters. So that's just another feature, and that is after the cell goes past it and is read, the flow cytometer can sort the cells into different tubes. And so it's a way of actually purifying specific cells from a population of cells. Yeah, so flow cytometry is a big deal. One of the things I mentioned about it is that it's especially used, because it's especially used for cells that are in liquid, it's used a lot in blood cell biology. And there's a lot of medical biology. Like, for example, you probably know that folks that have HIV have a reduced number of a certain type of immune cell. And that's really been able to be measured with the flow cytometer, like that. So there are a lot of flow cytometers used in medicine and in research. So one more person, and I marked also down Cameron. What's one thing your group talked about?
We were talking about the flow cytometer as well, but we were talking about the 2N and 4N distinction.
Yep. Yeah, so that N can be just thought of as numbers of genomes. That's not the original name for it, but that you can think of it. 2 genomes means moms and dads. And that's diploid, that's another term that we should learn. Diploid means 2. Ploidy also is like an N. Ploidy means number of genomes. So we can have 2N, 4N would be cells in G2. When we talk about a sperm or an egg, they're down to 1N or haploid. Because they're going to join together and make a fertilized egg that's diploid again. Yep. So questions people have about any of this stuff. Remember that one of the reasons we do a concept chat is the hope is that the first thing is that you recall, because that's the way you learn stuff, right? You're kind of practicing the song, right? I've been up here singing a little bit, and then you're like, I learned a verse, and I'm singing it right now, like that. So it's good to be aware of that nobody learns well by just walking away from it, right? I'm learning that song by playing it, and then just walking away. So it's really good after a class to just walk along and just, self-reflect on, what did you like, what did you learn, and can't learn it all, so you start out and try to build a framework of, what's the big picture items and stuff like that. So we're trying to get that done. Then another reason is because it's really helpful to have notes. You're building a resource that you can study with like that, and when it comes to a test, it's like a study guide. And you can see classmates' notes, and you're like, whoa, that student has pretty good notes, or, you know, better than mine, or not as good as mine, you know, and you can sort of see, maybe get some hints about how to, you know, take good notes, stuff like that. Yeah, other questions people have, and, because we are doing great and we have more time, yep? So we'll talk more about stuff, and we can ask any questions at all, and I'm remembering that we have to hand back your exams too, right at the end of class. All right, so next thing is that the research to learn about the molecular mechanisms of the cell cycle, that research was really started in about 1980, and there really were a couple of examples of scientists who made great progress, and they went on to win Nobel Prizes in chemistry. And one of the strategies was to try to learn about the cell cycle genetically. So let's look for mutations that are defective in the cell cycle. And those scientists studied the yeast that causes bread to rise, and that same yeast species is used in a lot of alcohol brewing. And then there's another yeast that's used in a lot of alcohol brewing, and they study that as well. And so the book talks a little bit about yeast. And those are eukaryotic cells, and they identified lots and lots of mutations in specific genes that were defective in the cell cycle. And they could use a flow cytometer to see at what point in the cell cycle did they stop. So they found some mutations that stopped in cells that were tetraploid. And lots of those mutations were in the genes for the proteins that regulate the G2 to M DNA damage checkpoint. Like that. And so they just named all those. So a genetic approach. Then some other scientists, and it was really just a total of just a handful of scientists that were working on this at that time, they were doing a biochemical approach. And they were looking at unfertilized eggs, because unfertilized eggs will mature, they become haploid, and then they wait. And it turns out that in different species, the egg cell waits at different points in the meiotic cell cycle. And then fertilization with a sperm causes them to move forward, like that. So it was a nice system where they could use different species and actually get cells arrested at different places naturally, and then stimulate them to go, right? So this will just show this, and we don't need to learn the details of the biology of the cells, the yeast cells, and so forth. Just the reality that it was just a big, awesome approach, because at some point the geneticists studying yeast connected with the biochemists studying frogs, and like a marine organism. And they're like, oh my gosh, we're identifying the same proteins. They were orthologs. Not just not just any orthologs, but orthologs that span all eukaryotes, just like that, right? Yep, so fantastic, and those people are still alive, and of course our author knew them and partied with them like that on his whole career, right? So they're in the book, because they're just like, oh my gosh, we don't need to worry about too much. We need to just focus on the reality of these are orthologs. Every protein that we'll talk about that's in the cell cycle is found in every animal on our planet, every plant on our planet, every single cell eukaryote. And we'll talk about these 2 points, the G2 to M transition, DNA damage checkpoint, and the G1 to S transition start control point, like that, okay? So in this chapter, we'll talk about these really 3 topics that will guide us through. So the first is that G2 to M transition, regulation of entry into mitosis. The second is the more complicated start. That's the one we've already been focusing on. That's what MIC regulates. That's the green light story. And then just the cell biology of mitosis. So yeah, during mitosis you can see the chromosomes, and you've learned this before in intro bio, but we want to go over it and add a little bit to it. So prophase, metaphase, anaphase, telophase, cytokinesis. Just a little bit about those, the cell biology of those stories, like that. And I think we got to stop. No, we're going to talk a little bit more, 2 minutes more. So here's just some of the biochemistry data that our book shows. And what it's showing is interphase, mitosis interphase, because it was a long time ago when scientists only understood that aspect of the cell cycle. You either were working on condensing the chromosomes and dividing the cell, or you were in between. And they found some proteins, biochemists did, that would go up and down with the cell cycle. And they thought those are probably part of the story of the cell cycle. And they called those cyclin proteins. So that's, again, really often biochemists are student-friendly scientists, you know, because they're like, yeah, they cycle, so we're going to call them cyclins. The geneticist, everything is just called CDC with a number, and it stands for cell division cycle mutants. You know, and you're like, that's not fun, right? Just a long list of numbers of genes. Yep. So the protein that controls the G to M transition is this protein, and it's called CDK1. And that stands for cyclin-dependent kinase. So we know what kinases do. They're enzymes that are going to phosphorylate targets. And when CDK1 is active, anytime the cell will go into M phase. And we'll learn why, because we'll learn about the targets of CDK1. And the targets are just that simple. It's biochemical, and it's got to be cause and effect, matter of fact. So let's deal with one right now. CDK1 is going to phosphorylate 2 helper kinases, and then those are going to go throughout the cell, and they're going to phosphorylate really specific targets. And one of their targets is histones. And we know what histones are. They're proteins that the DNA is wrapped around. And when histones get phosphorylated, they get packaged together tightly. So the specific reason why chromosomes become visible during mitosis is because the histones have been phosphorylated. And at the end of mitosis, at the end of M phase, the phosphates get removed, and the DNA decondenses and becomes invisible again. So that's the story. The biochemical story is if you activate that, you're going to do things like immediately start condensing your chromosomes. That should only happen if there's no DNA damage, so we need some control. And you can see it's called cyclin-dependent kinase. It needs to have a cyclin. So the whole role of the cyclins are just, they're needed for that kinase to be active. So they're one of the checks and balances. All right, now we have to stop. So I'm gonna hand back these, and you can head out, and there'll be a circled score at the end of your exam, and that's the number of points you got.